Okay, our first speaker is Hendrix. Noble's developed simulation and statistical models to understand how management decisions may affect fish and cetacean populations. He employs probability-based methods to express uncertainty and uses Bayesian estimates and Monte Carlo simulation for developing such models. He's the founder of, I believe it's pronounced QEDA Consulting, and he's an affiliate faculty at the School of Aquatics and Fisheries Sciences, the University of Washington, and he's based out of Seattle. So let's welcome Noble. Let's see if we have you unloaded here. Try to keep it 15 minutes.
Yeah. Okay. The button for the pointer is right in the top? Yeah, okay. Good shape. Okay, thanks. I just wanted to point out some of my collaborators on this project, Ray Hilborn, Craig Cunningham, who's a graduate student, Bob Lasardo at the University of Washington, and then a couple folks from the Northwest Fisheries Science Center, NOAA Fisheries. First of all, I'd like to acknowledge quite a few folks. This is a modeling exercise, and so as a result of that, I've collected essentially none of the data and have been piggybacking on all of the work. So this has been funded by the Delta Stewardship Council, and it's the early parts of this were funded by CalFed, and then in particular focused on today's topics. The bass information was provided by Department of Fish and Wildlife, in particular Marty Gingras. And so big thanks to him for all the information, and then there's been other sources of information provided from these other agencies. So the motivation for this model, this OBAN model, which stands for the Oncorhynchus Bayesian analysis, was to try and understand or try to develop a way of understanding how different factors that were hypothesized to affect winter run abundance, how those sort of played out relative to the population dynamics in our observed abundance estimates and observed juvenile abundance estimates. And so some of those factors were temperature in the spawning reaches and flows related to the timing of outmigration, conditions in the delta relative to the YOLO bypass and DCC, estimates of bass abundance, and then some nearshore ocean productivity indices and harvest. And so what we wanted to do is develop a modeling framework that had some specific objectives, and those were really to be able to fit and understand how these hypothesized factors were able to explain some dynamic vital rates, such as survival or reproductive rates. Then construct a statistical framework so that we could estimate the effects of those factors using these observed indices of abundance. And then where we could, we wanted to explicitly incorporate uncertainty in a Bayesian framework. And so that's what we started doing. So just to walk through the model structure, we basically have transitions between stages, different life history stages, here represented as Fry to Delta to the ocean. Those are modeled as a Beverton-Holt function, which has two parameters, P and a K, P being the productivity, or in our case, it's mostly survival rates, K being the capacity. And for those of you who are more graphically oriented, this is an example of a Beverton-Holt function. These are some data that we would potentially be fitting to, and it's a function of P, which is this survival rate in the absence of density dependence, and then K, which is our capacity, which then employs the density dependence function. And so these are occurring among these stage transitions. So then our full hierarchy, is to basically model these P's and K's as dynamic variables that are changing over time. In our case, they're essentially changing each year, and they're changing as a function of these factors that we're interested in evaluating. So that's what the X's are here. Those are the factors. These betas are the coefficients, and we're estimating this using logistic regression. That keeps our P in the appropriate range for survivals, essentially between 0 and 1. And then we can essentially model this. For the capacities, we also use a similar structure. The capacities have to be positive, so therefore we use log linear modeling where we log transform the capacities and make those a function of these hypothesized factors. So this gives us our framework that we can then use. This is essentially just a simulation framework, but when we go through the statistical fitting approach, then we can go about estimating these beta parameters, and that's what we're going to use to understand how important these factors are and base our inference off of these beta coefficients. So the first thing that we did was to develop a maximum likelihood modeling tool. We built this in software called ADMB, and it's auto differentiation model builder. And we built it's built a Visual Basic front end to help with model selection. And so what we were able to do is essentially build different models, alter the structure of the model by selecting different factors, estimate the fit to our, here's, for example, we have the log adult abundance, estimate the parameters by fitting, and then evaluate different models using Akaiki's information criteria. So, real briefly, some assumptions about the model. So these models, in a way, are very similar to multiple linear regression, only we're not assuming normality in the error structure, and we're not assuming linearity, for that matter. These Beaverton-Holt transitions are assuming assuming a nonlinear structure. However, we're still, the framework is somewhat similar. So as a result, some of the assumptions are we can identify relationships among these factors and the changes in abundance, and we can talk about them in terms of the amount of variability in the abundance that's being explained, but we're not really talking about any causal mechanism. We haven't really constructed the model with a specific causal mechanism involved. We then sort of take a second step at trying to explain some of why certain factors may be able to explain significant amount of variability versus other factors that may not. We have the issue of multicollinearity of factors, meaning that if you have two factors that are really highly correlated, then it's difficult to tell apart, which one is explaining. In our case, looking at both of the models that I'll show you, the multicollinearity is relatively low, sort of on the order of 0.6 or less. And then we have some distributional assumptions. We're assuming some log normal residuals with the fit to the data. And then finally, with the forecasting, this is a retrospective model, so any sort of large-scale changes to the system that were not captured historically are difficult to forecast because essentially this is tied into the historical nature of the system. Okay. So the first thing I'll do is show you some results from fitting to the Butte Creek spring run population data. And so for our model structure, we essentially have temperatures and flow in the fry stage. For the delta stage, of some interest here, we have BAS, which is a catch, estimates of catch that go back to prior to the 1970s. This model runs from the 70s through 2008. And then we have some proposed factors, upwelling, sea level height, sea surface temperature, and curl in the Gulf, and a harvest in the ocean. And here's the fit of our sort of best fit model. Indicating that, and again, this was fit in a maximum likelihood framework, so we're basically capturing sort of the nuances of the data, but there's obviously some data points that the model's not particularly sensitive to. Looking at the model structural uncertainty, what we see is sort of our selected model, the one with the lowest AIC score, contains BAS, curl, and sea level height, and then we have 3 or 4 of the top-ranked models here, and basically the top 3 account for about, using weights for about 85%. And so the coefficient values that we're interested in here is BAS in particular, and so we see that there's a negative relationship between, or our coefficient value is negative. For BAS, positive for curl, and negative for sea level height. So basically, you have higher abundances of spring Chinook when you have lower levels of striped bass abundance, when curl is positive, when sea level height is low for example, when these El Nino years tend not to be so great for spring run from Butte Creek. And then when exports are lower than average, this was the third model, but again, it's a negative coefficient value. Okay, so then the next level of modeling that we did really focused more on this Bayesian approach, and there's some real advantages to trying and using this same framework, but in a Bayesian approach. And so we use a software called WinBugs, which uses Markov chain Monte Carlo, or in some cases we use a distribution-free adaptive rejection step for those of you who have done some Bayesian estimation. So there's sort of this mix of samplers that are being used. In general, we used about a 50,000 iteration chain, and we did some, for 3 chains, the first half were thrown out, and then we used some convergence statistics basically to understand and make sure that these different chains that are starting in over-dispersed locations are converging to the same posterior distribution. So the model for winter run is composed of these 9 stages, eggs to alevin to fry, down through the delta, out to the bay and Gulf. And this is mimicking those factors that I had presented earlier. And then the indices of abundance that we're using or that we're fitting to are spawners from 1967 to 2008, and then also fry from 1995 on. And so, as I mentioned, 67, 2008, we have these annual escapements. We have some different measurement approaches or different measurement types that we're using over this time period. And so within the Bayesian framework, we can essentially set up some different measurement errors for these different time periods and allow the model to essentially sort out which were more accurate or which were less accurate. And then we have, like I mentioned, these juvenile production indices. So then some assumptions. We have some harvest rates that we needed to construct retrospectively, and we'll look into that. And then we have some maturation rates that we pulled out of Codaware tag analysis. So the covariates, we've got temperature for the alevins, this is basically temperature in spawning reaches. We've got minimum flows, which is a bend bridge, affecting the fry stage. Delta, we've got exports, YOLO access, and then the striped bass. And we've got 2 covariates, which I'll show you in a second. In the Gulf, we've got this upwelling index, curl, and then again, harvest in the ocean. So for striped bass, we've basically got 2 time series. We've got this catch time series, which is here in blue, and we've got the log. And these are log standardized covariate. So for all of the covariates in the model, they're standardized, or another way of saying it, they're basically using a z-score transformation. So they all have a mean of 0, and they have a standard deviation of 1. And the reason for doing that is you've got exports, which are on potentially a very large numerical range, and then you've got temperatures, which may be on a much smaller numerical range. And you don't want those numerical ranges to influence the model fitting, so you essentially make all of your covariates so that they have the same basic range, which is to have a mean of 0 and then a standard deviation of 1. So that's what's plotted here. Peterson estimates, so these are mark recapture estimates for age 3 plus, I think up to about 8. And so there's some gaps in the time series here. And so for an initial approach, we just used the straight log catch. And so when we fit, so here are the observed abundance indices for the escapement for winter run. Then the model is estimating these measurement error from the different time periods. Essentially, this middle period is the most or is imprecise, and it's due partly to the way in which these estimates were made due to some expansions of numbers after March 15th. And then we can fit the mean, so the model's fitting relatively well, and then these black lines are the 95% credible intervals. And essentially, the observed data are encompassed within these 95% intervals, with the exception of, I think, 1980 and 2000. So the model's doing a pretty decent job of reflecting the population dynamics. So we can look now at the posterior distributions of the model parameters. And so what's plotted here is in the black line is the posterior distribution. The red line is 0. And then this little number up here is the amount of the probability distribution that's less than 0. So you can see For temperature in the spawning reaches, you have 0.98 of this posterior probability density is less than 0. For exports, 0.96 is less than 0. For yellow bypass, 0.32. The one we're interested in right now is bass, and so we can see that about 0.18 is less than 0. So this suggests that that essentially that higher levels of bass would be associated with higher levels of winter run abundance. We can impute this abundance estimate because essentially we're fitting to catch data, and so the catch data may not be representative of the indices of abundance for striped bass. If we impute the abundance estimates by essentially regressing these two and using the errors, then we can also construct this model again, and essentially the results are almost the same, which is not surprising. Those two time series are correlated with about 0.8. Okay, so summary, basically we've got for winter run, lower temperatures, bass don't seem to have a big effect. You're standing up because little time. Okay, let me get to the end. Okay, so we've got a differential response to striped bass. Winter run are weakly related, and they're essentially in a direction opposite of what we might expect if there was a predation effect. Spring run seem to be negatively related. So one of the reasons, and again, this is getting to postulating potential mechanisms, given that we see some relationships in terms of the amount of variability being explained, that the Chinook abundance and timing of outmigration is quite different. So winter run is basically a small component, and they tend not to be, the timing tends not to be associated with the largest pulses of Chinook coming out, which are fall run, whereas spring run outmigration timing tends to be associated more towards the later spring, and so this may be explaining why we see some relationship with the catch data for striped bass. Finally, the metrics that I'm using are somewhat crude, and recent discussions with Marty Gingras suggest that there's probably other metrics that are more closely related to abundance. But ultimately, what we may be interested in is predation pressure, and by using catch potentially should reduce your adult abundance. And using the adult numbers may have a timing mismatch, because in fact, if you've got 2-year-olds and they're your main predators on juvenile Chinook because they're the largest component, then you'd be looking for a time mismatch with your adult abundance relative to, say, some sort of juvenile index. And so I really think it gets to having to have a better sense of what the population dynamics are for striped bass so that you can then use that to understand what the effects would be on the Chinook populations. And that's it for me. Yeah.
A couple minutes for questions.
Okay. Any questions? I know. It is the late. That's the afternoon.
Noble, I had a quick question.
Yeah.
I think I just might have missed a step. So you're modeling, the full life cycle, even when they're up in the Gulf. And did I hear right that you're including harvest as one of the predictors?
Yes.
So harvest
is one of the.
Is it all Gulf harvest, or are you able to apportion it to these stocks?
Oh, so it would be. So we use a harvest index that's. And this one was actually linked somewhat to harvest rates of winter run. And so essentially there's a Central Valley index that's focused on fall run, and so this was discounted due to management in the later time periods. But yeah, that's incorporated in those two ocean stages. Yeah. Okay.